Okay, this is chapter 12, and this is going to be the last chapter of the nervous system. So with the nervous system, now we're looking at sensory. We're going to be looking at special senses and general senses. So we remember that sensory receptors are throughout the body, and they're gaining information to send to the brain, to the CNS, so that our body can make decisions upon that information. These receptors link our nervous system to those changes, and they can be specialized by their cell or multicellular structures. General senses are ones that are receptors that are widely distributed throughout the body. So skin, organs, joints, there's general senses throughout those. Special senses, whenever you see special, when we're talking about nervous system, we're usually referring to the stuff in the head. So the eyes, ears, nose, and mouth. So in our body, we have these sensory receptors which are going to collect that information, and they go along the afferent or sensory fibers to the brain. The brain then has what's called sensation. It's a feeling that occurs when the brain becomes aware of that sensory impulse. Perception is a person's view of the stimulus, the way the brain interprets the information. This is an example of how sensation to perception can happen with an apple. It's interesting. So if you look at how looking at an apple, the sensory receptors of the nose will smell it, taste can taste it, and its path. All of these pathways for sight, hearing, information, and perception. There are some specific type of receptors that only do certain things, and the first one's a chemoreceptor. You see in its name that it's going to be chemical. This is respond to chemical concentrations. So we see chemoreceptors on our tongue, we see chemoreceptors in our blood vessels, places where you have chemicals, and the concentration of it is important. Pain receptors also have the name of nociceptors. They respond to chemicals released during tissue damage or pressure of some form that's causing pain. Thermoreceptors will respond to changes in temperature, so you see thermo meaning temperature. Mechanoreceptors are responding to a mechanical force, so touch, stretch, pressure. Those are all mechanoreceptors. And then photoreceptors will respond to light. So when you stimulate a receptor, it causes a local change in that receptor's potential, meaning it stimulates this receptor so that there's changes chemically and electrically in the receptor, so that electric current is now generating an intensity depending upon the stimulation. And so that receptor will then generate what's called an action potential. If it reaches a certain voltage in the neuron. This is all dependent upon the type of stimulus, and if it's strong enough to stimulate the receptor, thus the axon, which will generate an action potential, which I know we didn't cover in this semester, but an action potential is basically chemical and electrical current, in a sense, that travels down the axon only when certain factors are met. Now, if the receptor is part of the neuron, that receptor potential must be transferred to a neuron to trigger an action potential. If it's not part of a neuron. So if that receptor is connected to a neuron that isn't sending that information, it's going to try to connect to one that is sending that information up to the brain. Now, peripheral nerves will transmit their impulses to the CNS, where they then are interpreted and analyzed in the brain. Projection. This is when your brain projects where the source came from. So it helps you pinpoint a region of stimulation. Sensory adaptation is the ability to ignore an unimportant stimuli. So examples of this are your jewelry, your clothes, your undergarments. All of that is stimulating your nervous system, but it's not important to your brain, so it tunes it out. Sensory impulses will become less frequent and sometimes may cease, but if the stimulus is stronger, stronger stimulus is required to trigger impulses. So we have general senses. These are senses with small, widespread sensory receptors that are associated with our skin, muscles, joints, and viscera or organs. There are three groups. We have exteroceptive senses. These are senses associated with the body surface, such as touch, pressure, temperature, and pain. Interoceptive or visceroceptive senses. These are associated with the viscera or organs, such as blood pressure, stretching blood vessels. And then proprioceptive senses. These are senses associated with changes in muscle, tendon, and joints. So when you change position or exercise, your brain knows where your body is. There are three types of mechanoreceptors that provide touch and pressure. We have free nerve endings, which are common in your epithelial tissue. They're the simplest receptors, and they give a sense of itching. Tactile or Meissner's corpuscles. We saw this when we were talking about skin structures. This is abundant in hairless portions of your skin and lips. It detects fine touch and texture, and it distinguishes between 2 points on the skin. Lamellated or Pacinian corpuscles are large oval structures, and they're common in deep subcutaneous tissue, tendons, and ligaments, and therefore they're for detecting heavy pressure as well as a vibration. So here's a picture just like from our skin chapter. The free nerve endings at the top up here. We have our tactile or Meissner's corpuscle for touch, and then down deeper we have the lamellated or Pacinian corpuscles for pressure. Temperature receptors are called thermal receptors. In free nerve endings in skin, there are two types. There's warm receptors, which is sensitive to temperatures above 25 degrees Celsius or 77 Fahrenheit. Once you get beyond that, it becomes unresponsive to temperature. Oh, excuse me, not beyond 77, but once you get above 113 degrees, it's unresponsive to temperature, and now you're talking about pain. Receptors will kick in. Same with cold. It's sensitive to 50 degrees and 68 degree Fahrenheit. Below that, you start to get pain response, which will give you a freezing sensation or a burning sensation if it's too warm. So pain, again, those receptors are called nociceptors, and they consist of free nerve endings. They're widely distributed through your body. The nervous tissue of the brain lacks pain receptors, and it's stimulated by tissue damage, chemical, mechanical forces, or extremes in temperature. The thing that's interesting about these nociceptors is they do not adapt. If they do, it's very little. So this is basically for your protection. Now you can also have what's called referred pain, which is typically from your viscera. There's pain receptors in the viscera whose stimulation can produce a symptom or a sensation to the surface. So it may not be directly on the viscera, but it will refer its pain to other places. A common example would be a heart attack or heart tissue damage would cause pain, often in the left arm, left jaw, left side of the chest. Referral pain results from a common nerve pathway in which the sensory impulses from the organ and certain areas of the skin will synapse with the same neuron in the central nervous system. Here's a little map showing us referral pain, which is due to sensory impulses from two regions following a common nerve pathway to the brain. Here's another example how you can see the sensory nerve fiber coming from the heart and the pain receptor coming from the skin are going through the same pathway. So the regulation of pain pathways means how does it get to the brain? What happens? In the thalamus, remember we talked about that as our switchboard in the brain. So the thalamus is going to begin that sensation of pain. The cerebral cortex is really what's going to judge the intensity, the location, and produce any emotional or motor responses to that pain. Gray matter in the brainstem will regulate the flow of impulses from the spinal cord. And your body and your brain will actually produce some pain inhibiting chemicals called enkephalins, serotonin, and endorphins. Now, if you've ever heard of a runner's high, basically what that is after you've stimulated your muscles to a certain intensity and frequency, your body will start to release endorphins and enkephalins. That gives you this euphoric feeling. So that's an example of how your body uses those chemicals. Serotonin often is manipulated, I guess the best word is to explain it, when you take chemicals or drugs for depression, painkillers, those RA drug, like rheumatoid arthritis drugs. There's a bunch of them. If you ever listen to drug commercials and they'll explain it, they'll say a serotonin inhibitor or uptake inhibitor, stuff like that. That's what it's dealing with, is the serotonin, which is a pain inhibiting substance. Moving on to special senses. These are senses that have sensory receptors within large, complex sensory organs in the head. So smelling is coming from olfactory organs in the nasal cavity. Taste comes from taste buds in the oral cavity. Hearing and equilibrium is from the inner ears, and sight is from your eyes. Olfaction is the sense of smell, and those receptors are chemoreceptors. They respond to chemicals dissolved in liquids, and a sense of smell provides 75 to 80% of taste. So you've probably experienced having a cold before where your nose is congested, so you are not able to smell as well, so food does not taste as well. This is why, because most of your sense of taste comes from smelling it. The olfactory organs contain receptors, and they're bipolar neurons, and they're supporting epithelial cells. They cover the upper parts of the nasal cavity and portions of the nasal septum. Odorants may bind to almost 400 types of olfactory membrane receptors, and which will lead to that reaction where the nervous system is stimulated. Here is our olfactory bulb and olfactory nerve. This is on the inferior surface of the frontal lobe. And this is a sagittal view of the ethmoid bone. You can see here's the olfactory bulb. These are neurons that come down and they extend through the epithelial tissue there. Once the olfactory receptors are stimulated, nerve impulses travel through openings in that cribriform plate, which is this porous part of the ethmoid bone. That nerve sends signals to the olfactory bulb, then to the tract, and then to the limbic system for emotions, and then the olfactory cortex for interpretation. Notice in this order of nerve flow, you do not see thalamus. This is the only sensation, sensory information in the brain or in the body that does not go through the thalamus. Smelling goes straight to the limbic and then olfactory cortex. The olfactory bulb analyzes sensory impulses. The limbic system is the center for memory and emotion, and it provides emotional response to odorant molecules. Each olfactory receptor contains only one type of membrane protein, and so there's almost like a code to this as your brain will translate it. The olfactory organs located high in the nasal cavity above the pathway of inhaled air. They will undergo sensory adaptation very rapidly. The sense of smell drops by 50% within 1 second after stimulation. This is why you can smell something very strong initially, and then after a few seconds or so, you may not even smell it at all, or you've adapted to it that it's very minimal stimulation or smell. Olfactory receptor neurons are the only ones in direct contact with the environment, and they can be damaged. So if you are a smoker, this will damage your olfactory receptors. If you have to use nasal sprays all the time, this will create actual changes to the epithelium and damage your receptors. If you are somebody that sniffs something like drugs or paint or whatever, you can create damage, is the moral of the story here. Receptors are replaced regularly. It's very unusual for neurons to be replaced in a human adult. So these are ones that are an exception. Moving on to taste. The sense of taste is called gustation. The taste buds are the organs of taste. They're located on papillae of the tongue, the roof of the mouth, the linings of the cheek, and the walls of the pharynx. There's about 10,000 taste buds, each with 50 to 150 taste cells on them. The taste receptors are chemoreceptors. They are modified epithelial cells that function as receptors. There's little hairs that is a microvilli that protrude from those cells through pores of the taste buds, and they're the sensitive parts of your taste cells. Taste cells are replaced every 3 days. Here's some visual pictures of our papillae, which most of us refer to as taste buds, but really the taste buds are these tiny little cells that line the papillae. There are 5 primary taste sensations. We have sweet, stimulated by carbohydrates; sour, stimulated by acids; salty, stimulated by salts; bitter, stimulated by organic compounds; and umami, which is stimulated by amino acids and monosodium glutamate. Each flavor results in one primary taste sensation or a combination. Spicy foods can activate pain receptors, and taste receptors will undergo rapid adaptation. Its nerve pathway is a sensory impulse from those receptor cells traveling on fibers of 3 different cranial nerves. So we have facial, glossopharyngeal, and vagus nerve. The cranial nerves conduct impulses into the medulla oblongata. and then it goes to the thalamus, and from there it's interpreted in the gustatory cortex in the insula. Here is our projection pathway through the cranial nerves. Next is hearing. Our organ of hearing is the ear. There are three main parts: the outer or external ear, the middle ear, and the inner or internal ear. The outer part that you see is called the auricle. Another name for it is pinna. It's funnel shaped, and its intention is to collect sound waves. That sound wave then travels through a hole called the external acoustic meatus. If you were doing lab, you would see we learned this hole in the temporal bone in the skull. It's the same name. It's this opening that is going into the ear. It's an S-shaped tube. It is lined with ceruminous glands, which produce earwax, and it carries sound to the tympanic membrane that terminates at the tympanic membrane, or ends there. So you can see right here, here's your acoustic, external acoustic meatus. There's the tympanic membrane. That is what we would refer to as the eardrum, and it vibrates in response to the sound waves that are collected in there. So here is those items: the auricle, the tympanic membrane, the temporal bone that houses it all, and the external acoustic meatus. So before we talk about this, let's look at the middle ear. The parts of the middle ear are going to be the tympanic cavity, which, if I go backwards a second, you'll see is this space, this green space, behind the tympanic membrane. There are three tiny bones called auditory ossicles. They vibrate in response to the tympanic membrane, and their names are malleus, incus, and stapes. If you took any biology in high school or as a child, you probably learned them as hammer, anvil, and stirrup, but their medical names are malleus, incus, and stapes. They are then connected to a opening or a hole in the wall of the tympanic membrane, or tympanic cavity. This is called the oval window. The stapes will vibrate against it to move fluids in the inner ear. So here's the malleus, the incus, and the stapes. This is honestly not the best picture. They're pretty tiny, but they connect one after another. So in the line of order, if you go from the posterior or the back side of this tympanic membrane, the first bone to connect is the malleus, in the middle is the incus, and then the stapes is at the end, which connects to this coiled structure, and that's what's going to be pushing on the oval window to put the vibration into that inner ear. Here's a better picture. Here is tympanic membrane, the malleus, the incus, malleus, incus behind it, stapes right there. So the tympanic reflex is muscle contractions that occur during loud sounds. This is to help protect your ears, and it prevents damage to the hearing receptors. Muscles involved are the tensor tympani and the stapedius. We won't test you on these muscles, but just know what the tympanic reflex is. In the middle ear, we have the auditory tube, or otherwise called the eustachian tube, which is right here. This is a tube that connects your middle ear to your throat. It helps maintain pressure on both sides of the tympanic membrane. Remember, your tympanic membrane is somewhat exposed to the external environment, so there's atmospheric pressure pushing on it. So on the opposite side, we need to have a balance, and that's what that auditory tube does. By being connected to the nasopharynx area, you have like a valve-like flap in the throat that will cover it to keep it protected, but also to allow air to come and go from there to help maintain pressure on the tympanic membrane. You may notice pressure changes if you've ever been in an airplane, and you start chewing or swallowing, moving your jaw. That's to move that auditory tube to get that balance right in the ear. All right, the inner ear or internal ear. This is the big structure that's kind of funky looking. It looks like a snail. Well, there's two parts to it that there's so much detail to this structure. We're going to just kind of review the basic structures and a little bit about the function. We're not going to spend a lot of time in major detail on it, so this is why I'm going through this with you, so you know kind of what level you need to study it. So this inner ear has this this structure that, like I said, looks like a snail, but is covered on the outside by a osseous covering, which is like a bone. So it protects it. So if we did a cross section, this very kind of gray layer on the outside, that's the bony labyrinth. It's basically this bony canal. There's fluid then on the inside. So below the bony labyrinth, where it's brown, that is where you're going to have perilymph, which is a fluid. Then you have another tube inside of here, and that light purple tube is called the membranous labyrinth. This is a tube within here that carries more fluid called endolymph. So remember, if you think about how things, the suffixes of order, so peri is going to be more outside, endo is always the inner part, so endolymph is the fluid on the very inside. There's three parts of the labyrinth. We have the cochlea, which is this coiled portion, and that's functioning for hearing. And then these big loops are called the semicircular canals. They function in dynamic equilibrium. And then in the middle is an area called the vestibule, and that functions in static equilibrium. Dynamic equilibrium means moving. Static is sitting still. The cochlea is that spiral snail-shaped tube, and if we did a cross section, you would see little chambers, in a sense, and then connected with nerves to the cochlear nerve. Don't worry so much on the details of the parts of the cochlea. Let's look at the windows first. There are two membrane-covered windows. The oval window, which we talked about, where the stapes attaches to it and transfers vibration. That vibration is sent through the cochlea, and then any excess vibration that is left over will dissipate out the round window. So it comes in through the oval window and exits out of the round window at the bottom there. There are three compartments to the cochlea. The top part is called the scala vestibuli, and then the lower part is called the scala tympani. The cochlear duct is between them. So as the stapes here vibrates, it will send vibration through. If we elongate our cochlea, it will send it through the scala tympani and down through the scala, through the scala vestibuli and then down through the scala tympani. In our vestibular membrane, that is what's going to separate the scala vestibuli from the cochlear duct. Basically, it's a lining inside. There's a lot of tissue in here. There's nerve endings in here. There's some endolymph in here. This is where you're going to have your receptors for hearing. This is kind of what it looks like on a slide. So the spiral organ is also called the organ of Corti. It is the sense of hearing. It is sitting on the upper surface of the basilar membrane. has hair cells or receptor cells in there. And the tectoral membrane is like a roof over the hair cells, and it will move with vibration. And the vibration will cause the cilia or hair cells to contract and bend against that membrane. Different frequencies of vibration will move different parts of the basilar membrane, and nerve impulses will be generated. Sound reception occurs at the basilar membrane when it vibrates, and those hair cells are then putting them in contact with the tectoral membrane. If we straighten out the cochlea, you can see the receptor cells in different regions of the cochlear duct detect different frequencies of vibration. Our pathway is going up the vestibular cochlear nerve, through the medulla oblongata, the midbrain, the thalamus, and then to the auditory cortex. There's the vestibulocochlear nerve. Equilibrium is the second function of the ear, and we have two types. We have static equilibrium, which senses the position of your head when your body is not moving, and dynamic equilibrium, which senses rotation and movement of the head and body. Static equilibrium will be found in the area where the vestibule, which is basically this middle area. The structures that are part in here that are going to detect it are called the utricle and the saccule. Each of those will contain another organ called the macula, which is a patch of hair cells and supporting cells. The macula has hair cells embedded in a gelatinous material with these little crystals called otoliths on the surface. Gravity will pull the gel when the head changes position. Those otoliths will shift, and it will then bend the hairs in those receptors and tell the brain, going to the vestibular branch of the nerve, to the vestibulocochlear nerve, to the brain telling you just bent your head forward. Receptors for dynamic equilibrium are found in the three semicircular canals. Each canal contains an ampulla, which is a swelling, which is this rounded part here, which will communicate with the vestibule. The structure called the crista ampullaris is, again, hair cells in a gel-like structure, and here's what it looks like. It will move when your head moves and your body moves, and thus sending that information to the vestibular nerve, to the vestibulocochlear, and to the brain. Sight. Visual receptors are found in the eye. The accessory organs for the sense of sight are your eyelids, which are called palpebrae, eyelashes, lacrimal apparatus, which causes tears, and external eye muscles, which move your eye. Extrinsic eye muscles, excuse me. First, let's look at eyelids, or palpebrae. There's 4 layers. You have skin, muscle, connective tissue, and structure called conjunctiva. It's a mucous membrane that lines the eyelid and covers portions of the eyeball. You don't need to know the muscles that move the eyes, so you can kind of skip over the muscles in this section, or especially when we're talking about the eyes. There are some glands in the eyelids called tarsal glands that secrete oil onto the eyelashes for protection. The lacrimal apparatus is the lacrimal gland, which is up in the corner of your upper eyelid, secretes tears. That then will wash over the eye to keep it protected and wet, and then it will be collected in 2 ducts called the canaliculi. That then will drain into the lacrimal sac, which will then drain down into the nasolacrimal duct and into the nasal cavity. There is an antibacterial component of tears called lysozyme. Let's kind of go through the parts. Well, okay, so you can skip this page. We're not going to test you on the eye muscles. Let's look at the eye. There are 3 layers: an outer fibrous tunic, a middle vascular tunic, and an inner nervous tunic. The outer fibrous tunic is the white of the eye, called the sclera, and the front, which is transparent, called the cornea. The cornea helps focus light rays and transmits and refracts light. The sclera is white and opaque. It protects the eye, it attaches muscles, and it's pierced by the optic nerve and blood vessels on the posterior side. Here's the cornea and sclera. The vascular layer is the choroid coat, the ciliary body, and the iris. The choroid coat is the posterior 5-6, which is basically this layer on the under the sclera. It will contain melanocytes, and that will help absorb extra light. It also has blood vessels in there. Towards the front of the eye is a structure called the ciliary body, and this is the thickest portion of the tunic. And this is pigmented. It forms rings to hold the lens in shape, or hold the lens, basically. And then our iris is in front of that. That's anterior to the ciliary body, and this is in front of the lens. It's pigmented, and it controls the light entering the eye. The anterior cavity of the eye is between the cornea and the lens, and is filled with a watery fluid called aqueous humor. The lens is behind this, and this is a transparent biconvex structure that lies behind the iris. It's elastic, and it's kind of suspended or held in place by ligaments called suspensory ligaments. They attach to the ciliary body, and they move by the ciliary body pulling on them, and which will change the shape of the lens to help focus light rays, whether you're in close or long vision. Here's another picture of the ciliary body. This is that internal ring around the front of the eye, and this is helping contract and relax the lens, and the suspensory ligament will hold the lens into position. The lens lies just behind the iris and the pupil. Accommodation is basically the change in the shape of the lens to view close objects. The lens will thicken and become more convex when focusing on a close object, and then it thins and becomes flatter when focusing on distant objects. The iris, the color of your eye, is controlling the amount of light that comes in. It consists of connective tissue and smooth muscle. The pupil is the window or the opening of the iris, and dim light will stimulate the nerve muscle or the muscles, and the pupil will then dilate. Bright light will stimulate muscles and cause it to constrict. The amount and the distribution of melanin in the iris determines the eye color. The aqueous humor, again, is in the anterior and posterior chamber of the, but it's always kind of in the front here, so in the anterior chamber and then right behind the iris. The nervous tunic, or the inner tunic, is consistent of the retina. The retina contains visual receptors or photoreceptors. It's continuous with the optic nerve in the back of the eye, and it ends just behind the margin of the ciliary body toward the front of the eye. There's several layers to it. The macula lutea is a yellowish spot in the retina. Fovea centralis is the center of the macula lutea, and this is where you have your sharpest vision. The optic disc is just lateral to that, and that's a blind spot. It contains no visual receptors, and this is found where the nerve fibers from the retina leave the eye to become the optic nerve. All of this is held kind of flat against the choroid coat by a thick gel called the vitreous humor. So here's your retina in yellow, here's the optic disc, and the vitreous humor is filling all of that space to keep the retina attached to the choroid coat. The posterior cavity is a space enclosed by lens, ciliary body, and retina. It contains vitreous humor. There we go. See, there's the posterior cavity or vitreous humor. There's major groups of retinal neurons. The photoreceptors are bipolar cells and ganglion cells, and they provide pathways for impulses triggered by photoreceptors to reach the optic nerve. There's horizontal and amacrine cells, which will modify and integrate impulses. Refraction is bending of light, which occurs when light waves pass at an angle between mediums of different densities. A convex lens can cause wavelengths to converge, and a concave lens will cause light waves to diverge. So as light enters the eye, it is refracted by the convex surface of the cornea and the convex surface of the lens. So images focused on the retina are going to be upside down and reversed from left to right. It is your visual cortex in your brain that corrects that reversal and turns it the correct way. So really your eyeball is similar to a microscope in the sense that when the image comes in, it's upside down and backwards. Our photoreceptors are rods and cones, and rods are going to be the long, thin projection, so hence its name. Has a light-sensitive pigment called rhodopsin, and it's 100 times more sensitive to light than the cones are. It will provide vision in a dim light, produce colorless vision, and produce outlines of objects. Cones are short, blunt projections, which are light-sensitive to pigment. Pigment's called erythrolab, chlorolab, and cyanolab. Provides vision in bright light, produces sharp images, produces color vision, and in the fovea centralis, where you have your sharpest vision, that is only has cones in it. There's their shape. The pigments, again, rhodopsin is going to be light sensitive in pigment in rods, and it will decompose in the presence of light. The pigments in the cones, we have erythrolab, which responds to red light, chlorolab responds to green, and cyanolab responds to blue. Stereoscopic vision is providing a depth perception in a sense. So it gives you the ability to kind of look around an object, so we don't see everything as 2D. Our visual pathways are going from our optic nerve to the optic chiasm, and this is going to crisscross some of those fibers and then eventually get to the visual cortex of the occipital lobe. As we age, our senses will diminish. Hearing loss will be due to the damage to the hair cells in the spiral organ, degeneration of nerve pathways to the brain. Tinnitus is a ringing in the ears that can happen. Age-related visual problems including dry eyes, floaters, which is crystals in the vitreous humor, loss of elasticity of the cells or lens, so a decreased accommodation, glaucoma, cataracts, and macular degeneration are all examples of problems with your vision as you get older. Age-related smell and taste problems will be due to a lack of olfactory receptors. So that is chapter 12. Thank you very much.